All right, guys, so this week we are talking antibiotics and antibiotic resistance. Let's get into it. So chemotherapy is just a generic term that we use for any kind of chemical to treat a disease. Now, an antimicrobial would be any kind of chemotherapy that we use to kill microorganisms. And then there's a subset of those antimicrobials that are called antibiotics. And those are antimicrobials that are made by other microorganisms to kill other microorganisms, which sounds confusing, but basically we're just taking advantage of the fact that molds, funguses, bacteria, they've all been co-evolving together over forever. And so they've been learning how to attack each other and kill each other. And so we're basically using their weapons against them. Some antimicrobials are bacteriostatic, which means that they inhibit cell growth, and some of them are bactericidal, which means that they actually kill the cells. And it kind of depends on what you're trying to do, but for the most part, bactericidal is what you would want. Just a few of the highlights from antimicrobial history. Paul Ehrlich, we've talked about him before. He was a Jewish-German scientist. He developed one of the first chemotherapeutics, and that was called Arsphenamine. Now, Arsphenamine was the first commercial therapeutic, and it was for syphilis. Syphilis was a huge problem for hundreds and hundreds and hundreds of years, basically going back as far as history goes. And he won the Nobel Prize in 1908 for that. He did a ton of other stuff, too. But anyway, he was really, really important. And you can see arsphenamine, which is what he developed, is really, really interesting because it's using arsenic against other living things. And so, yes, that would be really toxic. That is true. But basically, humans could survive some level of arsenic, and the bacteria that you were trying to kill wouldn't. Now, another important contributor was Gerhard Domak. He was really important. He worked at Bayer, and his lab helped develop Prontosil, which was the first commercially available antimicrobial. And you can see Prontosil is here, and we're going to talk more about sulfa drugs and sulfonamides. And you can see on the right here, so that's one of the pieces of this sulfa drug. Prontosil will actually become the sulfa drug that will actually go on to inhibit the metabolic pathways for folate synthesis inside bacteria. Interesting guy. He was working before the Nazis took power in Germany, and then when they took power, they said, hey, you won the Nobel Prize, but the Nazi government was like, yeah, you're not accepting that because we're awful. And so he won the Nobel Prize, but he didn't get any of the prize money. So someone else that you may have heard of is Alexander Fleming. So Fleming, he was from Scotland, and he accidentally discovered Beta-Lactam antibiotics, which we're going to talk about today. So what happened was he had a bunch of plates, and his lab was kind of messy, and some mold got on his plates. It happens to everybody. Believe me, I get mold on our plates all the time. And he had some Staphylococcus growing there. And you can see here on this plate, there's a zone of inhibition around that antibiotic. And that's really interesting. So that means that the bacteria is not able to, at least not grow there. And it turns out it's actually being killed. And for that discovery, he shared the Nobel Prize in 1945 with two other people. Those two other people were Howard Florey and Ernst Chain. Florey was an Australian, but they were both working in London at the time. And what they were doing was Florey was inspired because he was working on penicillin, but they just couldn't figure out a good way to make enough of it to be useful. And there was this policeman, and he got a staph infection and was super, super sick. And he started getting better because Florey was giving him penicillin, but then they ran out of penicillin and the constable died. And so that shocked Florey so much that he was like, you know what, I am figuring out a way to make a bunch of penicillin. And Chain worked in the same lab, and he did a lot of the work of actually figuring out the structure of penicillin, which we're going to talk about later. And they figured out a way to mass produce it and purify it, and then it's estimated to save, I mean, this is a little old number, but 200 million lives. It's definitely higher than that. Now, there's a lot of history that we can't go into. I mean, there's a ton of just important things that have happened in the development of antibiotics. Something important to note, though, is that antibiotic resistance has existed almost in line with antibiotic development. So a lot of antibiotic resistances were discovered almost like the year or two after we started introducing antibiotics into the environment, which is really scary. And we're going to talk more about antibiotic resistance later after we talk about antibiotics. Now, as I mentioned before, the term antibiotic means it's derived from a microorganism, typically a fungus or from other bacteria. And so we have a few antibiotics. Some of these you may have heard of. So bacitracin is pretty common, polymyxin B, Ampho B, chloramphenicol we use in the lab all the time. I've used erythro, neo, strep, gentamicin. Those are all lab things. Penicillin's way at the bottom. But you can see these come from different species of either bacteria, either gram-positive rods or actinomyces, or some funguses at the bottom. And that's where the name of these molds comes from. So Penicillium mold is where we actually derived penicillin. And so we're going to talk about that. Now, you can have synthetic antibiotics. And basically what that means is we've discovered antibiotics from a lot of creatures. And then what we do is we do some organic chemistry, which we're not going to go super into today. But you can just change different groups and just change what's attached onto the main structure or the main functional component of an antibiotic. And then you can just change those things, and that can change, how reactive it is. It could change how long it stays in like the blood, whether or not it can go through the stomach lining, or you have to apply it IV, all kinds of different things. And we'll talk more about that later. But in the case of like this chart right here, you can see these are all different beta-lactam antibiotic classes. And you can see the basic class on the left, and they all, a bunch of these share similar structures. So like you got your penicillins and your cephalosporins, those all have beta-lactam rings. But you can see we can start to change like these R groups. And as we advance into further and further generations of antibiotic development, you can see we've gone from, just a straight up penicillin, like a penicillin G here with this nice ring. And then we start tacking more things on, and then we start tacking more things, and then we start adding more rings, and it just gets really, really complicated really fast. But hey, you know what's really scary? Bacteria will always develop antibiotic resistance to this stuff. So that's why we always have to keep developing these and only use them sparingly. So as we're talking about antibiotics, there's something that we need to think about. And this applies to lots of other things as well, lots of other chemotherapeutics, think cancers, where you need to make sure that the ideal characteristics of your drug are matching up at least as close as they can to what you really need. And there's some things that you may not think about. So one of the most important things is selective toxicity. That one actually makes a lot of sense. That's where you're actually trying to target whatever the disease-causing thing is and not the host. So in the case of a cancer, that can get kind of tricky because it's the host cells that are cancer. And so, if I use something to kill the cancer, well, it's the host's actual cells in the first place, so that might be toxic to the host. And so that's why a lot of chemotherapies are actually really, really toxic. You also want to be microbicidal, so you want to destroy the bacteria or cancer or whatever it is. I'm going to focus on bacteria. But you want to be microbicidal and not microbistatic. You want to actually kill it. Now, you also have to make sure that it's going to get where it needs to go. So it needs to be stable, especially in bodily fluids. There's all kinds of stuff floating around. And so you don't want it to get broken down prematurely. You don't want it to accidentally get excreted prematurely. You want whatever drug you have to have a decent half-life inside your host. You definitely want to try and avoid antimicrobial resistance. We'll talk about how to do that later. And you want to use something that's not going to damage the host, but you want to use something that's going to allow the host's immune system to actually defend itself. And that's where some microbistatic antibiotics might actually be decent if they can suppress the growth of a bacteria long enough to allow the host's defenses to actually kill it. How you actually deliver some kind of chemotherapy or drug or whatever it is really important too. So if you have a little kid, they don't know how to swallow pills, spoken from experience. And so they need a different way to be administered. They tend not to like shots either, but then again, I don't like shots. But that's something important to think about. Like, how am I actually going to deliver it? And is it reasonably priced? If I have to choose between a brand new antibiotic that's crazy expensive and one that's slightly cheaper, but it'll probably be about the same, then I'm probably going to choose the more reasonably priced one, which in the US healthcare system is a huge question that we have to ask. And of course, we're going to talk a little bit about this, but a lot of antibiotics can cause allergic reactions, or at least what's perceived as allergic reactions. And that's really important too. You don't want to kill your host. Jumping off that idea of selective toxicity is something I want to reiterate over and over again, which is that bacteria are pretty different from us eukaryotes. Our cells are pretty different. Now, we are cellular-based organisms, but we do know that bacteria and archaea and eukarya, we all have pretty big differences. And those differences are what we're going to take advantage of when we actually make antibiotics. And so if you get more closely related, so if we start looking at eukaryotic parasites like funguses, or we even get into bigger parasites like malaria or things like that, then what can happen is we have less tools to deal with those because there are less differences, at least on the molecular level, between a eukaryotic parasite and something like a bacteria. Now, viruses get really tricky because viruses do share a lot of the similar symptoms of disease, but they're not actively reproducing by themselves. They're using a host cell. And so if we're going to use an antiviral, which we'll talk about some antivirals today, we don't have as many tools because you can't directly use an antiviral on the virus itself. You sort of have to get into the cell that it's trying to take over and attack it there, which can get kind of tricky. So the next thing I want to talk about is the spectrum of antibiotics. And you can have narrow spectrum antibiotics and you can have broad spectrum antibiotics. And there's a lot going on in this chart, but basically all it's saying is, okay, at the top we have what kind of bacteria you're going to be dealing with. And of course, this isn't comprehensive, but some antibiotics are really, really specific. So like cefazolin would only work against gram-positive streptococcus bacteria. Okay, that's fine. But some antibiotics are really, really broad and they have broad spectrum. And so, for example, some of these you may have heard of, like tetracycline or tigecycline or gentamicin. Lots of these have multiple bacterial species that they can actually destroy. Now, in general, what you want to do is use the narrowest spectrum antibiotic that you possibly can use. Now, that could kind of depend on, your diagnosis. So a patient comes in, they have some kind of bacterial infection. Okay, let's put them on a broad spectrum antibiotic. We're going to quick do a gram stain. We see that, it's a gram-positive streptococcus. Okay, awesome. Let's throw them on some penicillin or some amoxicillin. And then once we actually figure out, okay, well, maybe we can narrow it down even further. And we can say, okay, we know it's not Clostridium. We know that it must be something else. Maybe it's some kind of Enterococcus. Okay, let's use trimethoprim because we know that's really specific. And so you narrow it down. Why is that important? Well, you don't want any off-target effects. So off-target effects are when you start killing off the normal microbiota that live on or inside of you, like on your skin, in your mouth, on your face, mostly in your gut is what we're usually thinking about. Because what can happen is you can develop a bacterial superinfection. Now, there are a couple different categories of superinfection, but the superinfection that's usually being talked about is from bacteria. And bacterial superinfections are when you start treating a patient with broad spectrum antibiotics to treat some like a staph infection or something, and you accidentally destroy all of their normal flora. And what's really important is that maintaining the bacterial flora in your gut is really, really important because they're taking up nutrients, they're taking up physical space, and they're going to kind of fend off their awesome place that they like to live, which is inside your intestines. And so if you kill them with some kind of broad spectrum antibiotic that you're trying to treat, somebody's got like an infection on their arm and you wipe out their gut bacteria, well, that's not good because you can have these second infections come in. And that happens a lot with Clostridium difficile or a C. diff infection, which can just shred your intestinal lining. It's really, really bad. So that's what this picture is. It's showing a histology of what happens only 30 hours after exposure. So C. difficile, Clostridium difficile, starts to secrete a special toxin that can just annihilate your intestinal lining. And so we're talking 36 hours after exposure. Only 36 hours. That's not that long. And you're going to start basically having such severe diarrhea that you could die. And that's really bad. And it's also super infectious. And so a lot of C. diff infections will happen in hospital settings too, because that's where people are put on really powerful antibiotics. And over here, these pictures are kind of gross, but this would be a C. diff infection inside someone's intestines. And so you can see it's pretty gross. Yeah, you don't want that happening. So I want to start talking about the different classes of antibiotics that exist. This class, I will not be doing a fully comprehensive going through every single type of antibiotic, but we're going to stick to the four main ways that they actually kill or cause a stasis of bacteria, and that's messing with their DNA or RNA, messing with their ribosomes with protein synthesis, messing with specific metabolic pathways, so that's like your sulfa drugs, as well as messing with cell wall synthesis, which is the first one we're going to talk about. That's beta-lactams. Now, there are other classes, of course, but that's generally the four big ways that antibiotics are going to actually kill bacteria. Now you can see on this really cool chart all the different structures. There's a lot of structural variety that occurs in antibiotics, but you can see we've had a lot of these around for a long time. Some of them, like in the light green, are bacteriostatic, and some of them in the darker green are bactericidal, and generally you want your bactericidals. Why do you want a bactericidal? Well, you want to completely eliminate the bacteria that's, causing disease. And so this chart, it's super simple, but basically, you add your antimicrobial, and if it's bactericidal, then you're hopefully killing it off, and then you can, take the person off the antimicrobial. But if you do that too early with like a bacteriostatic drug, and the body hasn't been able to mount an adaptive immune response effectively until that point, well, then the bacteria could just take over and just start doing its thing, which that's not good. And so in general, you want bactericidal antibiotics. So let's get into the specific mechanisms of how we're going to address antibiotics. And the 4 different ways, I'm going to go through these in this order. And we're going to start with cell wall synthesis, which is going to be our beta-lactam antibiotics. And then we're going to move on to the protein synthesis inhibitors. And so that's going to be your aminoglycosides, your tetracyclines. That's one that a lot of people have heard of. And then we're going to go on to nucleic acid synthesis inhibitors. So those are like your quinolones and your fluoroquinolones. And then we'll end with the sulfa drugs. So let's start talking about cell wall synthesis. Now, this is one that you've probably heard of before. So these are your penicillins or your beta-lactam rings. So your beta-lactam antibiotics is the better term for it. So these are antibiotics that have a beta-lactam ring. So a lactam ring is just this nice square structure. You can see it keeps showing up right here. It's incredibly reactive because of the ring strain. So that square should make you feel really uncomfortable because squares don't normally exist in nature and in bonding patterns because it's a lot of strain to actually make a 90-degree bond. But that's what actually makes penicillins and other beta-lactam antibiotics, which there's tons of those, it's what actually makes them really, really effective at grabbing onto the active site of an enzyme or a set of enzymes called penicillin-binding proteins. And they're called that because that's how we first discovered them. We co-localized penicillin with this enzyme inside of a bacteria. And we were like, okay, that's really weird. Let's call it a penicillin binding protein because it's grabbing onto penicillin. Well, it turns out what it is it's the enzyme and set of enzymes that actually build peptidoglycan, which we've talked about before, right? So gram-positive bacteria have a really thick layer of peptidoglycan, and gram-negative bacteria have a really thin layer of peptidoglycan, but they both have peptidoglycan. And so you can use a beta-lactam antibiotic to destroy those enzymes, or at least disable those enzymes. And so you can see here in the top right corner, I've got a couple different beta-lactam antibiotics. There are tons of different ones. So sometimes we use penicillin with modified R groups on it, but there are lots of generations of penicillins that exist. And basically what's happening is they're acting as direct inhibitors. They look a lot like the precursor that the penicillin binding protein, so it wants to build peptidoglycan, and it's going to grab a bunch of these precursors, which are peptido, so that's peptides, so that's like amino acids, and glycans, which is sugars. And so it's going to grab onto those and then use that to build the structure of peptidoglycan. It's like laying little bricks down. Well, these kind of look like bricks, so you can see there's some similarities here, especially like this corner of that lactam ring. And so the beta-lactam ring fits in that little corner. And so it reacts then with the active site and it disables the active site. And so that's how penicillin binding proteins are disabled.
Structurally, most bacteria consist of a cell membrane surrounded by a cell wall, and for some bacteria, an additional outer layer. Internal to the cell membrane is the cytoplasm, which contains ribosomes, a nuclear region, and in some cases, granules and/or vesicles. Depending on the bacterial species, a number of different external structures may be found, such as a capsule, flagella, and pili. In gram-negative bacteria, the gap between the cell membrane and the cell wall is known as the periplasmic space. Most gram-positive bacteria do not possess a periplasmic space, but have only periplasm, where metabolic digestion occurs and new cell peptidoglycan is attached. Peptidoglycan, the most important component of the cell wall, is a polymer made of N-acetylmuramic acid alternating with N-acetylglucosamine, which are cross-linked by chains of 4 amino acids. The function of the bacterial cell wall is to maintain the characteristic shape of the organism and to prevent the bacterium from bursting when fluid flows into the organism by osmosis. Synthesis of the peptidoglycan and ultimately the bacterial cell wall occurs in a number of stages. One of the first stages is the addition of 5 amino acids to N-acetylmuramic acid. Next, N-acetylglucosamine is added to the N-acetylmuramic acid to form a precursor of peptidoglycan. This peptidoglycan precursor is then transported across the cell membrane to a cell wall acceptor in the periplasm. Once in the periplasm, the peptidoglycan precursors bind to cell wall acceptors and undergo extensive cross-linking. 2 major enzymes are involved in cross-linking: transpeptidase and D-alanine carboxypeptidase. These enzymes are also known as penicillin-binding proteins because of their ability to bind to penicillins and cephalosporins. Eventually, several layers of peptidoglycan are formed, all of which are cross-linked to create the cell wall. Gram-positive bacteria may have more layers than gram-negative bacteria and thus have a much thicker cell wall. Beta-Lactam antibiotics include all penicillins and cephalosporins that contain a chemical structure called a beta-lactam ring. This structure is capable of binding to the enzymes that cross-link peptidoglycans. Beta-lactams interfere with cross-linking by binding to transpeptidase and D-alanine carboxypeptidase enzymes, thus preventing bacterial cell wall synthesis. By inhibiting cell wall synthesis, the bacterial cell is damaged. Gram-positive bacteria have a high internal osmotic pressure. Without a normal rigid cell wall, these cells burst when subjected to the low osmotic pressure of their surrounding environment. As well, the antibiotic penicillin-binding protein complex stimulates the release of autolysins that are capable of digesting the existing cell wall. Beta-lactam antibiotics are therefore considered bactericidal agents.
So that was a video from Mechanisms in Medicine. They're a little bit older videos, but they're super, super well done. I'm going to be showing a few of those because they address exactly what's happening, give you a nice visual of what's going on. Now, those videos also have a second portion that talks about resistance. And I'm going to kind of be progressively talking about different antibacterial resistance mechanisms as we go, because they can kind of explain a little bit more about how these antibiotics actually work. And so speaking of beta-lactams, one of the ways that beta-lactam resistance is engendered is by using a special enzyme called beta-lactamase, because ASE is enzymes, right? And so what it's going to do is it decarboxylates, which is just a term for basically breaking this bond right here, which is what it would normally do if it was trying to bind into that active site. And so the beta-lactamase just sort of triggers the beta-lactam ring early. And so it goes around looking for beta-lactam antibiotics, and it finds it, and it cuts that little ring. And so if that ring is cut, then it can no longer react with the active site. And now you have disabled the antibiotic, and now you are a resistant bacteria.
Bacterial resistance to beta-lactam antibiotics may be acquired by several routes. One of the most important mechanisms is through a process known as transformation. During transformation, chromosomal genes are transferred from one bacterium to another. When a bacterium containing a resistance gene dies, naked DNA is released into the surrounding environment. If a bacterium of sufficient similarity to the dead one is in the vicinity, it will be able to uptake the naked DNA containing the resistance gene. Once inside the bacterium, the resistance gene may be transferred from the naked DNA to the chromosome of the host bacteria by a process known as homologous transformation. Over time, the bacterium may acquire enough of these resistance genes to result in a remodeling of the segment of the host DNA. If this remodeled DNA segment codes for cross-linking enzymes, i.e. penicillin-binding proteins, the result is the production of altered penicillin-binding proteins. These altered penicillin-binding proteins can still cross-link the peptidoglycan layers of the cell wall, but have a reduced affinity for beta-lactam antibiotics, thus rendering the bacterium resistant to the effects of penicillin and other beta-lactam agents. This transfer process has resulted in penicillin-resistant S. pneumoniae through the acquisition of genes from other naturally occurring penicillin-resistant Streptococcus species. A second important mechanism by which bacteria become resistant to beta-lactam antibiotics is by the production of enzymes capable of inactivating or modifying the drug before it has a chance to exert its effect on the bacteria. Depending on the bacterial species, the gene coding for these enzymes may be found as part of the host DNA or on plasmids, which are small, self-replicating units of genetic material. Bacteria are capable of passing these resistance plasmids to each other by conjugation. When two bacteria come into close contact with each other, a small channel is created between them which allows one of the bacteria to pass a copy of the resistance plasmid to the other. If the plasmid is transcribed and translated, the bacteria will begin to produce inactivating enzymes. These enzymes, capable of destroying beta-lactam antibiotics, are known as beta-lactamases. In gram-positive bacteria, the beta-lactamase enzyme is generally inducible, resulting in a large amount of enzyme being produced in the presence of the drug. In gram-negative bacteria, the beta-lactam enzymes are produced constitutively, i.e., even when the antibiotic is not present. Gram-positive bacteria release the beta-lactamase enzyme from the cell into the extracellular environment where it inactivates the drug before it enters the bacterial cell. In contrast, gram-negative bacteria retain the beta-lactamase enzyme within the periplasmic space, resulting in a more efficient mechanism than gram-positive bacteria. Ultimately, the destruction of the beta-lactam ring of the antibiotic renders it incapable of binding to the penicillin-binding protein, and thus the bacteria become resistant to that drug or class of drugs.
Now, beta-lactamases are not the only way that you could have beta-lactam resistance, but I'm going to kind of just address various ways of resistance development over each topic that I'm going to hit. And then after we talk about all of these different things, then I will come back and we'll talk about them a little bit more. And so that's going to bring us to our next type of inhibition, which is protein synthesis inhibition. So protein synthesis inhibition, we know that ribosomes are a really cool little organelle. It's kind of an organelle, right? And we know that ribosomes are responsible for making proteins. They do the process of translation. So they take a messenger RNA and they use a transfer RNA right here. That's this funny looking thing. And they use that to actually attach on a chain of amino acids. And then it keeps on growing. And there are lots of different classes of antibiotics that affect different portions of the ribosome. So you can see here some of these, like tetracyclines, they actually block the tRNA, which is sort of bridging the gap between amino acids and RNA, and messenger RNA. And so it blocks that right there. Streptomycin is a little bit different. So instead of directly binding onto something, the streptomycin can actually indirectly bind to this 30S portion. So that's the smaller chunk of a prokaryotic ribosome, and it's able to change how that looks, and now your messenger RNA doesn't fit quite right, and so it can't actually do the translation with this tRNA. Chloramphenicals are a little bit different, and so what they do is they prevent the bonds from being formed between peptides. So that's kind of interesting, and so there's lots of different ways that we can actually inhibit protein synthesis. Now, this next video, before it gets into the antibiotics and how they actually affect protein synthesis, it's going to cover protein synthesis a little bit, but it's just a really good background to have under our belts.
The DNA is shown as a circular double strand within the bacterial cell. Like the DNA of all living organisms, it contains the unique genetic code for all of the proteins required for bacterial survival. These include the proteins required for reproduction, growth, repair, and regulation of metabolism. It also codes for the 3 kinds of RNA that are essential for carrying out protein synthesis. These are known as ribosomal RNA, rRNA, messenger RNA, mRNA, and transfer RNA, tRNA. In order for the bacteria to begin protein synthesis, the double-stranded DNA molecule must first unwind and separate in the region which codes for the specific protein that is to be made. Only one strand of the DNA serves as a template for this process known as transcription. Transcription results in the formation of messenger RNA, mRNA, which is a mirror copy of the DNA segment. Once the strand of mRNA is complete, it will detach from the DNA template and in turn become attached to ribosomes. Bacterial ribosomes are made of a small 30S and a large 50S subunit. After the 2 subunits join together around the strand of mRNA, synthesis of the polypeptide chain begins. This step involves the aligning of transfer RNA, tRNA molecules, and sequence along the mRNA. Each tRNA carries a unique amino acid determined by the sequence of the tRNA, which, when aligned along the mRNA and ribosome, join together to form the polypeptide chain. This step is known as translation. The ribosome will continue to add amino acids to the growing polypeptide chain until it reaches a point along the mRNA that signals it to stop. At this point, it releases the finished protein molecule. Macrolide antibiotics, such as erythromycin, act as inhibitors of protein synthesis by attaching to the 50S ribosomal subunit. As a result, they block the ability of the ribosome to synthesize the polypeptide chain. By inhibiting protein synthesis, macrolides are considered bacteriostatic antibiotics. However, at higher concentrations and with lower bacterial density or during rapid bacterial growth, macrolides may be bactericidal.
So that's going to bring us to another mechanism of antibiotic resistance, which is called an E-Flux Pump. So influx would be coming in, efflux would be leaving. And so an E-Flux Pump is able to expel compounds typically against a gradient. Because what's going to be happening is you can imagine you have a bacteria and it's in some kind of liquid, let's say, I don't know, your intestines or your bloodstream or something, and you take some kind of antibiotic, and now there's tons of antibiotics outside the cell and not very many inside, and they start leaking in through a variety of ways, either through specific channels, they could come through kind of generic porins, but basically they're getting inside the cell. This would be a gram-negative bacteria because it has periplasmic space here. But if those drugs are inside, they're going to cause damage. And so protein synthesis inhibitors aren't necessarily the only drugs that do this, but I'm just kind of using that as a jumping off point. And so E-Flux pumps can be really, really specific, or they can be generic. And we see E-Flux pumps not just in bacteria, but we also see them in like cancers. So there are lots of cancers that can have multi-drug specific E-Flux pumps, and they're actually able to push out anti-cancer drugs. And so I want to show the second half of that protein antibiotic video, which talks about E-Flux pumps.
Changes or modifications to the 50S ribosomal subunit, i.e. the target binding site for macrolide antibiotics, will confer resistance to macrolides and sometimes other classes of antibiotics. This type of resistance may be of a high level. This mechanism of resistance is mediated by the erythromycin ribosome methylation gene, which is found on plasmids, or transposons, i.e. small genetic elements which are capable of moving from one bacterium to another and integrating into the host chromosomal DNA. Copies of the gene are transported to other bacteria via plasmids or transposons through pili-like channels. The gene is incorporated into the new bacterial genome. During the process of protein synthesis, this bacterium will transcribe and translate the genetic code of the gene, resulting in the production of a protein enzyme capable of methylating the 50S ribosomal subunit at a specific position. This altered 50S subunit results in decreased binding affinity for macrolides and other antibiotics such as lincosamides and streptogrammin type B. This pattern of resistance is referred to as the MLS phenotype. Because the macrolide antibiotic is unable to bind to the 50S ribosomal subunit, it is unable to inhibit protein synthesis, and thus the bacteria itself is not harmed, continuing to produce polypeptide chains of amino acids. A second mechanism of bacterial resistance to macrolide antibiotics is mediated by E-Flux pumps. These E-Flux pumps are encoded by the MEFA gene, which is a transposable element. Because they confer resistance to only macrolides and not lincosamides or streptogrammin B, they are referred to as the M phenotype. The E-Flux pumps are energy dependent and, for S. pneumoniae, result in moderate levels of resistance. These pumps traverse the cell membrane of the bacteria and function to pump out the macrolide antibiotic after it has entered the bacterium. It should be noted that for other bacteria, such as Staphylococcus aureus, for example, a different efflux system, which is plasmid-mediated and encoded by the MSRA gene, results in macrolide resistance as well as lincosamide and streptogrammin resistance in some cases. Despite the presence of these efflux pumps, macrolide antibiotics continue to enter the bacteria. However, once inside the cytoplasm of the bacteria, these efflux pumps actively remove the macrolide antibiotics before they have a chance to reach their target, the 50S ribosomal subunit, and bacterial protein synthesis is unaffected.
Let's get into nucleic acid synthesis inhibition. Now, that's typically how you're going to target like RNA polymerases, which viruses and bacteria, they have their own RNA polymerases, or you could go after DNA replication in like a bacteria. So they use a machine called topoisomerase, which our cells do that too when we undergo replication. And it's targeting that. Basically what topoisomerase does, and we'll talk more about this when we talk about genetics, is it fixes one of the problems that can occur during replication. So our cells undergo mitosis, bacteria undergo binary fission, but when they replicate their DNA, it can get kind of tangly in topoisomerase, or in the case of bacteria, it's called DNA gyrase, but they have the same job. And basically they fix that problem. And so quinolones and fluoroquinolones can block that. And quinolones are a really good example of adjusting your R groups. And so you can see in this bottom left corner picture here, you can change your R groups, and depending on what you actually put in each of those positions can have a huge impact on what you're actually doing. Like you can change if you're targeting gram positive, you can change if you're targeting gram negative. If you throw a fluorine on here, fluorine is a really electronegative, a really reactive atom, and sticking that on here actually helps make it more sensitive to gram negative bacteria. Also on this R group here, most of your quinolones are going to have a triangle right here in this position, and that's super reactive. If you thought a square was reactive, talk about a little triangle there of atoms. That's going to react like crazy, and so it increases the overall potency, or in this case, like how reactive actually is it? How well does it actually disable topoisomerases?
In this animation, we demonstrate the biology of DNA replication, leading to bacterial cell division in a gram-positive bacterium, such as S. pneumoniae. The DNA is shown as a circular double strand within the bacterial cell. Like the DNA of all living organisms, it contains the unique genetic code for all of the proteins required for bacterial survival. Bacteria replicate by a process known as binary fission, whereby one bacterium separates into two new daughter cells. However, before this can occur, the bacterium must make an identical copy of its complete circular DNA. DNA replication requires that the two strands of DNA separate so that the genetic code of the bacterium can be read and a new complementary strand can be created for each of the original strands. To accomplish this, various enzymes, known as helicases, break the hydrogen bonds between the bases in the two DNA strands, unwind the strands from each other, and stabilize the exposed single strands, preventing them from joining back together. The points at which the two strands of DNA separate to allow replication of DNA are known as replication forks. The enzyme's DNA polymerase then moves along each strand of DNA behind each replication fork, synthesizing new DNA strands, in red, complementary to the original ones. As the replication forks move forward, positive superhelical twists in the DNA begin to accumulate ahead of them. In order for DNA replication to continue, these superhelical twists must be removed. The bacterial enzyme, DNA gyrase, which is also known as Topoisomerase II, is responsible for removing the positive superhelical twists so that DNA replications can proceed. DNA gyrase is an essential bacterial enzyme composed of 2A and 2B subunits, which are products of the GYRA and GYRB genes. This enzyme has other important functions which affect the initiation of DNA replication and transcription of many genes. With the combined involvement of these enzymes, an entire duplicate copy of the bacterial genome is produced as the two replication forks move in opposite directions around the circular DNA genome. Eventually, as the two replication forks meet, two new complete chromosomes have been made, each consisting of one old and one new strand of DNA. This is referred to as semi-conservative replication. In order to allow the two new interlinked chromosomes to come apart, another bacterial enzyme is needed, which is known as Topoisomerase IV. This enzyme is structurally related to DNA gyrase and is coded for by the PARC and PARE genes. Topoisomerase IV allows for the two new interlinked chromosomes to separate so that they can be segregated into two new daughter bacterial cells. This animation will demonstrate two mechanisms of fluoroquinolone action. Fluoroquinolone antibiotics, shown here, are synthetic molecules that are bactericidal. The potency of these drugs is greatly improved by the addition of a fluorine molecule at position 6, and thus the term fluoroquinolones. Fluoroquinolones rapidly inhibit bacterial DNA synthesis, resulting in bacterial cell death. Although much is known about the molecular events that underlie the action of quinolone antibiotics, much remains to be clarified. Fluoroquinolones act by inhibiting the activity of both the DNA gyrase and the topoisomerase IV enzymes. For most gram-negative bacteria, DNA gyrase is the primary fluoroquinolone target. Fluoroquinolones have been shown to bind specifically to the complex of DNA gyrase and DNA rather than the DNA gyrase alone. As a result of this binding, quinolones appear to stabilize the enzyme DNA complexes, which in turn results in breaks in the DNA that are fatal to the bacterium. A second mechanism of fluoroquinolone action is shown here. With some exceptions, topoisomerase IV is the primary target of fluoroquinolone action in most gram-positive bacteria, such as Staphylococci and Streptococci, with DNA gyrase being a secondary target. The separation of two new interlinked daughter strands of circular DNA is disrupted. The final result on the bacteria, however, is the same. Bacterial replication is disrupted and the bacterium breaks apart. The relative potency of different fluoroquinolone antibiotics, and thus their spectrum of activity, is dependent in part on their affinity for either DNA gyrase or topoisomerase IV, or both.
So we're covering lots of different kinds of resistance as well, and quinolones in particular can have tons of different ways, and all of these antibiotics, there's lots of different ways that bacteria can avoid them. But this one in particular, I wanted to talk about target alterations. And so if you have your gyrase or your topoisomerase inside of a bacteria, then this would be like its structure here of its active site. And then this is your quinolone or your fluoroquinolone binding in here. And if you change this active site's shape, which some of the other antibiotics we've already talked about do this as well, especially in those videos. But in quinolone resistance, that's definitely a way that bacteria can use to resist quinolones. And so in this next video, they're going to just show a little bit of that.
One of the most common mechanisms by which bacteria acquire resistance to fluoroquinolones is by spontaneously occurring mutations in chromosomal genes that alter the target enzymes, DNA gyrase, and topoisomerase IV, or both. The frequency with which these spontaneous mutations occurs may be in the range of 10 to the power of -6. The effect of mutations on the activity of an individual fluoroquinolone will vary depending on the number of mutations, the location of the mutations, and which target enzyme is affected. If a mutation occurs, either in the GYRA or GYRB gene, that alters DNA gyrase and results in a reduced affinity of the fluoroquinolone antibiotic for this enzyme, the organism will become resistant. Similarly, a mutation may occur that alters topoisomerase IV, either in the PARC or PARE gene, and results in a reduced affinity of the fluoroquinolone antibiotic for this enzyme, and the bacterial organism will become resistant. It is important to note that for some fluoroquinolones that have similar affinity and potency against both target enzymes, mutations in both DNA gyrase and topoisomerase IV will be needed for resistance to occur. In resistant organisms, semi-conservative replication continues.
The next set of antibiotics I want to talk about are the metabolics. So these are your sulfa drugs, which I mentioned before. So your sulfonylamides, so they look like another molecule. So in this case, they look like PABA, which is para-aminobenzoic acid. It's a precursor for a bunch of things. Basically, in your hydrofolic acid synthesis pathways, which humans do use that as well. So you can get some off-target effects by inhibiting the folic acid synthesis. So folic acid, so you may know, like if you're pregnant, you should take lots of folate or folic acid. So that's where this is coming from. So it's that same pathway. And basically you just need that as a precursor to make nucleotides, some proteins as well, but usually we think about it as targeting nucleotides. And so this sulfa drug looks like that precursor. And so you can see it can bind in here and block the enzyme's ability to actually use PABA to generate these downstream effectors. And there's other drugs too, like trimethoprim is a really common one, and often sulfonamides or sulfonamides and trimethoprim are used in conjunction because they're both inhibiting this pathway. I don't have a video because they just didn't have a really good video for this one, but the concept is there. It's just blocking this pathway. Now, I've talked about a bunch of different resistance mechanisms, just to kind of hit them again and talk about a couple other ones. So E-Flux Pumps, they're just pushing the antibiotic back out. You could just change your surface, so either they can't get through the porins that you have, or they can't get through the cell wall anymore the way that it was able to before. You could use an enzyme, so that would be like Beta-Lactamase targeting things. There's lots of versions of those against all kinds of different antibiotics. You can change the shape of the target itself, or you can try and go through a different pathway. So with the sulfa drugs, what sometimes bacteria can do is they can find an alternative way to actually generate the end product. So they can still make tetrahydrofolate or something, but they can do it a different way or using a different set of enzymes. So something else we need to talk about are the bacterial side effects. We're not going to go super in depth. Of course, you could have a secondary infection or a bacterial super infection occur using an antibiotic. It just wipes out the normal flora, and then like a C. diff infection can swoop in. But there are other immune-related side effects that can happen from certain antibiotics. A lot of people actually are not truly allergic. So an allergy would be, stimulating degranulation of a mast cell and releasing histamine and causing what you would typically associate with an allergic reaction, swelling, which is edema, typical things that you would think of with an allergic reaction. There are some things that are technically, they look like an allergic reaction like that, but they're not technically immune related, or they're not technically from how you would normally associate an allergic response. So like it might not be antibody or T cell mediated. It might be messing with some kind of pathway, which is kind of what's going on in the middle here. But you can have degranulation of your mast cells releasing histamine from certain medications, and that can happen in some people. So in this case, this guy's got super swollen face and super swollen lips because his penicillin has caused an edema, which that's never a good thing. And so we probably shouldn't put him on penicillins. But actually, a lot of people who think they have penicillin allergies don't technically have penicillin allergies. They're just having mild reactivities. So we've hit a lot of bacterial antibiotics, but we should talk about some of the other ones. So I'm going to quickly go through all the other different subsets. So we've got our antifungal drugs. So basically, remember, one of the keys is that we're looking for things that are different between whatever's invading you and your own cells. And so we have to look at what's different. Well, funguses they do look really different, but they are eukaryotes, and so they have a lot of similarities. And so we have to say, okay, well, what's actually different about them? Well, they have ergosterol instead of cholesterol, and so that's one way that we could target them. So we can use drugs that target the enzymes that generate ergosterol because they're kind of different from what your cells would do to make cholesterol. And there are a few other pathways as well. But basically, we have a few antifungal drugs that we can use. But for the most part, we're really just trying to target these random things that look kind of different. And the same goes for antiparasitic drugs. So these would be bigger eukaryotes. So your single-celled ones like malaria, schistosomiasis, toxoplasmosis, things like that. And basically what you're looking for is what's different. Well, these cells aren't super different from your own cells, and so we have to kind of try and attack them in different ways. And a lot of times we can do that because they have an alternative life cycle, and so they'll go between like a larval stage and an egg stage or something like that, and so we can try and target it there. So some of these I've seen before, like ivermectin causes a chlorine increase, permethrin causes a sodium increase, and so you're just trying to target these things specifically that are different enough so that you're not killing the host, because that would defeat the entire purpose. There are a lot of antiviral drugs as well. So my background is HIV, which is on the right side here. And when we talk about viruses, we'll focus in on HIV in particular because there's lots of really interesting drugs. But on the left here are specific drugs that are designed to target coronavirus. And you can see there's a lot of variety here. But basically, you're either trying to target the entry, so can I just block it completely from getting inside the cell? Or when viruses actually leave the cell, most of the eukaryotic viruses that we're dealing with need to steal some of the plasma membrane with them and then kind of cut themselves away and float away. And so we can block them that way. We prevent that cutting, basically. Other times we're trying to target things inside the cell that have to do with their life cycle. So the drug has to be able to get into the host cell. That's important. But also it needs to be able to mess with something inside the host cell that doesn't actually kill the host cell, but does kill the virus, or at least stop its reproduction. And so a lot of times you can have things that target the enzymes that come from the virus that are different from us. So like HIV has reverse transcriptase as an enzyme. Well, humans don't have that. And so that enzyme is something that we can target, either with a nucleotide or a non-nucleoside inhibitor. And they have their own proteases that are very specific, and they have their own integrases, which our cells don't have that. And so those are just things that we can target. Now, the last thing I want to talk about is antibiotic resistance. Antibiotic resistance I've been talking about the whole time, but basically they've always existed because antibiotics by definition come from biological systems, right? So we're talking about like Penicillium mold makes penicillin. It makes Beta-Lactam antibiotics, and it's been fighting bacteria for as long as they've been alive on this planet. And so you can just assume that a bacteria has dealt with a fungus in the past, and the same with a fungus. A fungus has dealt with bacteria in the past, and they've been using these chemical weapons against each other for basically before humans were even here. And so bacteria and funguses, every kind of microbe is used to these selective pressures that we can apply. And biological systems will always evolve to resist a selective pressure. And basically, once you put that selective pressure on a biological system, they will start to evolve away. So assume that 99.9% of a virus is susceptible to some kind of antiviral drug. Well, the problem is that 0.1%, and that 0.1% will then continue to live on. Now, what's kind of tricky is that there's usually some kind of fitness cost to those mutations that allow them to be resistant. And so you can see on this graph in the bottom left here, when we go from our mixture of viruses, some are just randomly able to resist whatever drug treatment we're trying to put on them. This could be a bacteria as well. And so you get a resistant set of quasi-species over here, but they may not be able to actually live as well as these other ones over here that we were able to kill. And so there's a fitness cost. So maybe they don't grow as fast, or maybe they're not as resistant to some other kind of antibiotic. But it doesn't really matter because they're resisting whatever this selective pressure is at the time. And so that's why, especially like with HIV or now with antibiotics, we've started using multi-drug therapy. So instead of just giving one person a single antibiotic, like, okay, we're just going to put them on penicillin, we say, okay, no, we need to make sure that we kill every possible one, because the odds of a bacteria being resistant to a penicillin, a cephalosporin, a quinolone, and some other kind of drug are really, really low, that you would get something that's resistant to all four of those, as opposed to just one by itself. And so we call that multi-drug treatment. Antibiotic resistance is a huge problem, and it's estimated to kill a lot of people, like a ton of people, in the very, very near future. And so there are a lot of complexities in how we actually deal with that and how we need to approach that. And so to end this lecture, I want to show this TED Ed video that goes into some of those subtleties.
Antibiotics. Behind the scenes, they enable much of modern medicine. We use them to cure infectious diseases, but also to safely facilitate everything from surgery to chemotherapy to organ transplants. Without antibiotics, even routine medical procedures can lead to life-threatening infections, and we're at risk of losing them. Antibiotics are chemicals that prevent the growth of bacteria. Unfortunately, some bacteria have become resistant to all currently available antibiotics. At the same time, we've stopped discovering new ones. Still, there's hope that we can get ahead of the problem. But first, how did we get into this situation? The first widely used antibiotic was penicillin, discovered in 1928 by Alexander Fleming. In his 1945 Nobel Prize acceptance speech, Fleming warned that bacterial resistance had the potential to ruin the miracle of antibiotics. He was right. In the 1940s and 50s, resistant bacteria already began to appear. From then until the 1980s, pharmaceutical companies countered the problem of resistance by discovering many new antibiotics. At first, this was a highly successful and highly profitable enterprise. Over time, a couple things changed. Newly discovered antibiotics were often only effective for a narrow spectrum of infections, whereas the first ones had been broadly applicable. This isn't a problem in itself, but it does mean that fewer doses of these drugs could be sold, making them less profitable. In the early days, antibiotics were heavily overprescribed, including for viral infections they had no effect on. Scrutiny around prescriptions increased, which is good, but also lowered sales. At the same time, companies began to develop more drugs that are taken over a patient's lifetime, like blood pressure and cholesterol medications, and later, antidepressants and anti-anxiety medications. Because they are taken indefinitely, these drugs are more profitable. By the mid-1980s, no new chemical classes of antibiotics were discovered, but bacteria continued to acquire resistance and pass it along by sharing genetic information between individual bacteria and even across species. Now, bacteria that are resistant to many antibiotics are common, and increasingly, some strains are resistant to all our current drugs. So, what can we do about this? We need to control the use of existing antibiotics, create new ones, combat resistance to new and existing drugs, and find new ways to fight bacterial infections. The largest consumer of antibiotics is agriculture, which uses antibiotics not only to treat infections, but to promote the growth of food animals. Using large volumes of antibiotics increases the bacteria's exposure to the antibiotics, and therefore their opportunity to develop resistance. Many bacteria that are common in animals, like Salmonella, can also infect humans, and drug-resistant versions can pass to us through the food chain and spread through international trade and travel networks. In terms of finding new antibiotics, nature offers the most promising new compounds. Organisms like other microbes and fungi have evolved over millions of years to live in competitive environments, meaning they often contain antibiotic compounds to give them a survival advantage over certain bacteria. We can also package antibiotics with molecules that inhibit resistance. One way bacteria develop resistance is through proteins of their own that degrade the drug. By packaging the antibiotic with molecules that block the degraders, the antibiotic can do its job. Phages, viruses that attack bacteria but don't affect humans, are one promising new avenue to combat bacterial infections. Developing vaccines for common infections, meanwhile, can help prevent disease in the first place. The biggest challenge to all these approaches is funding, which is woefully inadequate across the globe. Antibiotics are so unprofitable that many large pharmaceutical companies have stopped trying to develop them. Meanwhile, smaller companies that successfully bring new antibiotics to market often still go bankrupt, like the American startup Achaogen. New therapeutic techniques like phages and vaccines face the same fundamental problem as traditional antibiotics. If they're working well, they're used just once, which makes it difficult to make money. And to successfully counteract resistance in the long term, we'll need to use new antibiotics sparingly, lowering the profits for their creators even further. One possible solution is to shift profits away from the volume of antibiotics sold. For example, the United Kingdom is testing a model where healthcare providers purchase antibiotic subscriptions. While governments are looking for ways to incentivize antibiotic development, these programs are still in the early stages. Countries around the world will need to do much more, but with enough investment in antibiotic development and controlled use of our current drugs, we can still get ahead of resistance. The world is full of accidental inventions that have had major impacts on our lives. The potato chip, the microwave oven, Post-it notes. But did you know that the first antibiotic was invented by accident? Has any mistake in human history saved more lives? Learn more about this fascinating story with this video.